Okay, let's start with the Golgi apparatus. First, a short definition. It is found within the cytoplasm of both plant and animal cell. It is composed of stack of membranes. In the plant cell, this stack of membranes is called the dictosome. It is discovered in 1897 by the Italian biologist Camillo Golgi, and that is why it is named by him. It is also called Golgi complex, or Golgi complex, and this is how it looks photoed by a microscope, but this is how we will found in our textbook, because it is much easier to study it like that. Now, its functions. Its main function is to take the not ready for use proteins from the raw endoplasmic reticulum and to pack and mark them in order they to be ready to go out of the cell or remain into the cell and be used later. And that is why it creates lysosomes and transports lipids around the cell. This is the summarized process. Like there are the proteins, they go inside, and some of them are packed to go outside. Some of them remain inside and become lysosomes. Okay, very easy to remember function, but it is very important, because if we have a disease, because disease caused by him can be very fatal. Like the Alzheimer's disease, it attacks the brain and acts like dementia. Its symptoms get worse over time. For example, at the beginning, only our head is hurting, and we can remember some things, but maybe at some time we cannot remember what happened before 1 hour. And badly, it is not cured, but there are treatments and drugs which make it be not so fatal. Another not so famous disease is the inclusion cell disease, or also called eye cell. An interesting fact about the Golgi apparatus is that it disappears when a cell is going to divide and reappears in the new two independent cells, who are the divisions of the big cell. The point to remember, it is found only in eukaryotic cells, not like the ribosomes, which can be found in non-eukaryotic cells.
Hello, 92, it's Kirill. I'm going to explain you what are the ribosomes and what's their function in the cell. The ribosomes are small particles of protein and ribonucleic acid, or in other words, RNA, which can be found in the cytoplasm of the cells. The function of the ribosomes is to convert the information that the DNA sends into protein. In other words, the ribosomes are little factories in the cell that listen to the orders of the boss, the DNA. Now, after you know what are the ribosomes, I'll explain about the ribonucleic acid. The RNA has a lot in common with the DNA, but there are three main differences between them. Firstly, the sugar in the RNA is ribose instead of the deoxyribose. Secondly, RNA is generally single-stranded and not double-stranded. And finally, RNA has uracil instead of the thymine. In fact, we can compare the DNA and RNA with the boss and workers. The DNA has all the information of the cell, so it's the boss of the factory. The RNA, or the workers, also has copies of this information, And based on this information, proteins are made up in the factory, aka ribosomes. There are three types of RNA, which determine the function of each one of them. The messenger RNA, or just mRNA, has molecules that carry the information from the DNA to the other parts of the cell. Ribosomes contain two subunits. These subunits are made up of several ribosomal RNA molecules and as many as 80 different proteins. And the last type is the transfer RNA. Its function is to bring the code from the mRNA's amino acids to build protein and encode it. That's all from me, and now I'll let Nicole explain the translation.
Translation. Ribosomes use the sequence of nucleotide bases in an mRNA molecule to produce polypeptide by joining amino acids. After this process, the polypeptide folds into its final shape or connects with other polypeptides to become a protein. The ribosomes do this process. This process is called translation of an mRNA message into protein. Steps in translation. Transcription doesn't take part in the translation, but it's critical to it. The translation can happen when there is transcribed mRNA. First, in an eukaryotic cell, the process happens in the nucleus. After the transcribed mRNA goes into the cytoplasm, the translation is carried out by the ribosomes. Everything begins when a ribosome attaches to an mRNA. The amino acids come from the tRNA when the codon passes through the ribosome. Then these amino acids are attached to the growing chain by the ribosomes. The amino acids in each tRNA are just from one kind. In the tRNA there are 3 anticodons that are supplemented with one mRNA codon. When there is a methionine, the anticodon is UAC, and it pairs with the methionine codon AUG. For the next codon, there is another binding site in the ribosome. If the next codon is UUC, AAG anticodon fits against it. The amino acid is brought into the ribosome by the second tRNA molecule. The ribosomes help the first and the second amino acids form a peptide bond. After that, this bond is broken and the mRNA moves into third binding site from where it exits the ribosome. Then the ribosome moves to the third codon where the tRNA brings the amino acid. The polypeptide chain stops growing when the ribosome reaches top codon. When this happens, it releases both the newly formed polypeptide and an mRNA molecule, completing the process of translation.